Hello everyone, welcome back to another class on regeneration biology. And in today's class, we will try to learn some data that is generated in my lab, and I will try to explain this to you. So the roles of histone deacetylases in regeneration of vertebrates like fishes and amphibians. So we will touch upon mainly fish fin and amphibian tail and limb. So there is also some several papers that are published in this field from our lab and also from other labs. And in general, there is a discrepancy between the regenerative ability between mammals and fishes when it comes to histone deacetylase. In simple sense, I can tell you histone deacetylases, if inhibited in zebrafish, it is not favoring regeneration. Whereas in mammals, if you inhibit, it favors regeneration. So that much day and night difference is there, but we will see. But the mechanism is not fully understood. All we know is you do it, you get the result. But let us try to learn what is the mechanism probably acting behind it through these results. So in this picture, you are seeing stages in zebrafish fin and axolotl limb regeneration. And you can see this in several textbooks, etc. Procedure is simple. Take a fin, zebrafish fin, amputate it, and you have a wound epidermis, and you will start seeing the blastema that is being formed by around 2 days time, and the regenerative outgrowth happens completely. And same with the limb also. You cut it, you have a wound epidermis formation just like fin, and the innervation is necessary. We saw in previous classes already. And you have an apical ectodermal cap being formed, and underneath there is a blastema that is formed, and the new limb patterning happens, and the limb gets regenerated fully, as you can see in this picture. Simple. But what is the take of, or what is the role of epigenome modifiers or epigenetic modifiers, and how they intercalate into the mechanism of regeneration? We will see them. So after amputation, zebrafish fin, after amputation, you dip the fish in trichostatin A, which is an histone deacetylase inhibitor. All family of HDACs will be blocked with trichostatin A. You do not get regeneration. We know as long as you keep the fish in TSA, you will not get fin regeneration at all. But what we did is slightly different. What we did was took a normal fish, exposed it to trichostatin A. You blocked HDACs and then amputated, and did not put TSA or did not block the HDAC after amputation. We wanted to see what is the role. One of our earlier study, we have shown that if you withdraw it in retina, what we have shown is if you block the HDACs with the TSA, and after that you withdraw it, the regeneration continues. After, say, if the regeneration should have completed by 2 weeks of time, it takes 3 or 4 weeks time, but regeneration happens. But here, what we have seen is, if you block 3 days prior to amputation, as you can see, that is written -3, -3, -2, -1, and 0 day you amputate it, and then put the fish in just water, and then collected at 4 dpa. You compare it to the control. In 4-day post-amputated scenario, this much blastema you are supposed to get it. Trichostatin 0.1 micromolar, 0.25 micromolar, and 0.5 micromolar. There is a increasing dose concentration. In the highest dose you get, compared to the control, you get minimal blastema. So what it tells you, even if you block the HDAC much before the amputation, you do not get efficient regenerative response. And same scenario you get it for axolotl tail and limb. Both of them you see it. So the procedure is same, -3 days to 0 day, and then put HFR solution is a normal solution in which the axolotl is kept. So this is pre-cut scenario, and this is 0 dpa. But in this pre-cut scenario, animal is exposed to TSA. Here it is DMSO, whereas in here it is TSA. That you keep in mind. And after 0 dpa, 7 dpa, you are supposed to have this much blastema formed in DMSO, whereas in 2 micromolar concentration, you do not get proper tail blastema. Same with limb also. Same axolotl procedure is same. You amputate it, you are supposed to get this smooth blastema in the stump that is formed. But here in TSA treated, you get a sharp rudimentary blastema, which is not powerful enough to cause a adequate regenerative response in a given amount of time compared to that of control. So if you look, we followed it up to 56 days in a normal same pre-treatment scenario. DMSO treated one, you can see pre-cut, cut 8 day, 12 day, 16 day, 20 day, 24 day, 28 day, 32 day, 56 day. What you are seeing is by 56 day, the limb is somewhat similar to that of the pre-cut scenario you end up getting. But when you put two different concentration of TSA, one is 1 micromolar, another is 2 micromolar, you see the blastema formed is very slow and eventually it forms. And same with the 2 micromolar also, blastema formed is slow and eventually it forms. So if you look carefully, say here by around 24 day, the digit formation starts, 24 day post amputation in control scenario. Whereas in 1 micromolar, the same digit formation starts in 28 day. Because 24 day, you don't see anything. Same way 2 micromolar, you see there is still blastema is there. And even 28 day digits hasn't formed. 32 days, the digits has formed. So there is a 24 day, 28 day, 32 day equality is there. That means there is a delay, same like what you saw in retina. There is a delay in that experiment was different, in which where you injure the retina, put them in TSA, no regeneration, and then they withdraw the drug. Here we have done the TSA treatment beforehand, and there is a reason behind it. We wanted to know what changes in the homeostasis of the animal takes place that prevents the blastema formation. We are more interested in knowing the role of HDAC inhibition before amputation. We want to delink the role of HDAC in post-amputated scenario. So that is why we exposed in a pre-amputated scenario. So eventually in a delayed process, like actually the limb can get in around 32 days, you can get it, but 56 days been shown because we wanted to have a comparison with the rest of them. So by 56 days, they all look the same. But in 2 micromolar concentration, the regeneration is a slow process. Now, we looked what are the possible enhancement that occurred. So one of the candidate we looked by looking into literature, we saw that there is a HDACs have a good control over the collagen gene expression. So we looked zebrafish caudal fin and also axolotl tail and axolotl limb. We looked for the levels of collagen 4 and also looked at the levels of histone 3, lysine 27 acetylation. Normally, histone deacetylases are supposed to remove the acetyl group from the histone proteins and also various other proteins, but we are interested in the histone proteins, especially histone 3, lysine 27 residue. If acetyl group is there, the chromatin can be very loose and gene expression can happen. If acetyl group is removed, that will make that moiety, lysine moiety, vulnerable for methylation and the chromatin can become very tight. In any ways, if you block the HDACs, then the acetyl group on this histone will stay back. That is what you can see. The acetyl compared to the DMSO, the acetyl group is very strongly present, and that is why increasing concentration of TSA increases the H3K27 acetylation level, and GAPDH is the control. Interesting to note, the collagen 4 levels are increasing because of TSA pre-treatment. Remember, these are all no amputation. See, this is the regime, -3 to 0 day. Although you amputated it, you are not waiting for any regenerative response. So technically, in uninjured condition, just because of the TSA treatment, you have got increased levels of collagen 4 and increased levels of lysine H3K27 levels. Same thing is seen with axolotl tail and axolotl limb. So what you learn from here is that the pre-treatment of TSA or the prior to amputation, if you block the histone deacetylases, you end up having too much of collagen 4, We have discussed collagens are part of ECM, extracellular matrix protein, and these ones are super abundant. That is what you should understand. The matrix proteins are super abundant in the fin, tail or limb because of histone deacetylase being blocked. Same thing is confirmed by immunofluorescence level, not just Western. Zebrafish caudal fin 0 dpa, axolotl tail 0 dpa and axolotl limb 0 dpa, but they all have received TSA pre-treatment. You can see the green color collagen 4 expression is very strong, 0.5 micromolar, 1 micromolar of TSA, the levels are high, and this is showing the DAPI, which is the nuclear strain, and this is the merge. You can also see in the axolotl tail there is an increased levels of collagen 4, and you can see this is the DMSO level. This should have been the actual level, but because of the TSA treatment you have an increase in level. Same here, 2 concentrations of TSA is been tried in the limb. You are having an increase in the levels of collagen 4 because of TSA treatment or the blocking the HDAC. Now we thought, if collagen deposit is the culprit, if collagen deposit is to be blamed, then what if you get rid of the excess collagen? So one enzyme that we can use is for this purpose is collagenase. So what we did, we took these fishes which are pre-treated, and the pre-treatment increased the collagen levels. So we also gave the collagenase enzyme in the blastema. Although the blastema forms very little, we gave collagenase enzyme in the blastema and we could rescue the effect. See, you can see TSA treatment 0.5 and 1 micromolar blastema level is very low. This yellow dotted line, if you look, you can know the blastema levels are very low. However, because of the collagenase treatment like 0.05, 0.5 and 5 milligram per milliliter, that is the collagen concentration we have used, we can see it rescued the TSA mediated inhibition. This should have been the blastema levels, and you could get into this in the highest concentration. Same way 1 micromolar, this should have been the blastema level, but we could rescue it. Interesting thing to note is in the normal DMSO one, where there is no collagen deposit. Normal collagen is there, but there is no excessive deposit. There, when you remove the collagen, you see a reduced blastema. So what you understand from here, the collagen is necessary at a threshold level. Too high also problem, too less also problem. It's like, again, I give an example of the salt used in food. If you put 0 salt, food will not taste good. If you put too much of salt, food will not taste good. But there is a threshold, adjacent level. If salt is less in the food, putting salt, it will make it tasty. If salt is more in the food, getting rid of salt by whatever mechanism, getting rid of salt will make the food tasty. Same logic you should keep in mind. It's not that collagenase is a favoring enzyme. If collagen is a troublesome molecule, then collagenase is a favoring enzyme. When collagenase is not a troublesome molecule, then collagenase will do harm. So that is what you get from the DMSO treatment. Now, same result we can get it by collagenase treatment that rescues the TSA phenotype in the limb and also in the tail of axolotl. So you can see the DMSO, how the collagenase treatment is reducing the blastema in the DMSO, whereas in TSA the reduced blastema, as you can see here, there is a reduced blastema that is present in the PBS treated. No collagenase, you don't have any rescue now, so you get a reduced blastema, but that will bounce back because of collagenase treatment. Same thing we can see in the zebrafish, not zebrafish, in the axolotl tail also. In the PBS, you have got a reduced blastema formation, but if you give collagenase, you can rescue and the blastema bounce back. So from this, what you understand, because of the TSA treatment or because of the inhibition of histone deacetylases, the excessive collagen is to be blamed for lack of or the delayed regeneration. So if you can prevent or you can reduce that excessive collagen by collagenase treatment, then the trouble is alleviated. So this is the information we gain from here. We then performed an RNA sequence analysis. That is, we looked for the entire ECM genes. Collagen is one of the ECM genes. So if you look for what is the general take of the ECM genes because of histone deacetylase inhibition, what we found that these genes are upregulation and these are all the downregulation. This is a gene enrichment analysis what has been done, and this whole group is for the extracellular matrix genes, and the procedure is same, -3 to 0 day. And 0 day we have two things we have done it. One is 0 day RNA-seq and another is 3 day RNA-seq. So what we found that there is an upregulation of collagen or the collagen family extracellular matrix genes in the because of the TSA treatment compared to the DMSO. TSA in general upregulates extracellular matrix genes, and these levels and the downregulated genes are less. Upregulated genes are more, which belongs to the extracellular matrix family. But there are some ECM proteins which, not that if there are 100 ECM proteins in an animal, all 100 out of 100 is upregulated. Some are downregulated, but the majority are upregulated in 3-DPA. So we have done also 0-DPA, but due to time limitation, I cannot go and analyze all of them. 3-DPA has been used here. Then we did a heat map representation of the RNA-seq data, which shows the collagen as well as MMP genes. So we looked what is the role of this collagen. If collagen has to be increased, the heat map, you can see the red color, so many collagen family genes are there. Collagen is not just a collagen 4, there are plenty of other collagens also. You can see a bunch of collagen genes in 3-DPA, which should have been down because of which favors the regenerative response. But you have seen in TSA treated sample, because of the HDAC inhibition, you see a large number of collagen family that is gone up in red in color. And concomitantly, we can also think why collagen genes will go up, or even if collagen genes go up, we have shown by Western and immunofluorescence also collagen 4 levels are high. So we thought probably there is a possibility that the degrading molecule like matrix metalloproteases, they may have an opposite trend. So we saw because of the collagenase, because of the HDAC inhibition, along with the collagen protein levels going up, their transcription going up, protein going up, also some of the metalloproteases levels are going down. That means already transcription of collagen is high, translation is happening, but their degradation also is now prevented because many of the MMP proteins are downregulated. When we looked further the RNA-seq data to understand some other angle, what is that angle? We looked for the immunofluorescence data of collagen, gave us some idea that similar to these excessive deposit of collagens, that is creating a huge matrix of extracellular deposition. This may interfere with some cell migration into that site, which may be delaying. It is not stopping, it is delaying. It's like you have more hurdles. Then we thought, can this interfere somehow with some immune response because of this TSA treatment or because of HDAC inhibition? To our surprise, what we saw, compared to the actual scenario where there is no treatment, there is a decrease in the immune response genes in that injured site. So if you pre-treat the animal with the TSA, the immune response genes are downregulated. As you can see in this gene expression or gene ontology analysis, you can see this red color defense response, response to biotic stimuli, and lot of this highlighted red color highlighted genes are downregulated, which are occurring because of TSA treatment. And we also found something and this has been done both in 0 day and also 3-DPA. So both scenario, because of the TSA treatment or after post injury, we have seen some of the interesting immune response genes are downregulated. We looked further. Can we make an extra plot further on looking for candidate genes? So far in the previous class we have learned pro-inflammatory cytokines are needed soon after injury, which should be taken over by anti-inflammatory mechanism. Both are needed. So we looked for the levels of pro-inflammatory cytokines and anti-inflammatory cytokine. What we saw, because of the TSA treatment in both 0 DPA and 3 DPA, because of TSA treatment, we saw there is a decrease in the anti-inflammatory. That means the environment remains pro-inflammatory. Then we looked, what about pro-inflammatory? Because of the TSA treatment, we saw there is an increase in the pro-inflammatory gene expression events, as you can see here. So pro-inflammatory scenario remains for a longer time and the anti-inflammatory scenario doesn't kickstart. So this, it's just like you are earning less money, but the income increased also. Something like that. This is what happening here. Anti-inflammatory cytokines levels go down and the pro-inflammatory cytokine levels go up. These together probably could have contributed to the delayed regenerative response. We also have discussed in the previous classes that in mice, many scenarios where the pro-inflammation is favored or the pro-inflammation is facilitated, the regeneration is not welcome there, or regeneration cannot happen effectively. Now we looked into another mechanism that is specifically looked for the regulatory T cells. Regulatory T cells are important. One of the next class we will be discussing about regulatory T cells. But understand, regulatory T cells are one of the earlier granulocytes, which are CD4 positive cells, and they're important. They are very much important to have a normal regenerative response. So we looked these caudal fin of the zebrafish and looked what is the level of this CD4 positive that are FOXP3 positive. FOXP3 positive is an indicator of regulatory T cell. They are in short form, they are known as Tregs. We saw that in both 0 HPA and 3 HPA, hours post amputation caudal fin, the levels in TSA treatment, the Tregs, that is regulatory T cells levels are decreasing, and probably that could account for lack of or the delayed regeneration because of TSA treatment. And what we looked further was, of course, we all know soon after injury there has to be a free radical mechanism, there is a reactive oxygen species kicked in, etc., which is necessary for the immune cells attraction. So we looked, can we mimic the scenario by treating either with TSA or simply put LPS. What is LPS? Bacteria lipopolysaccharide. When bacteria lipopolysaccharide is deposited in the injury site, then pro-inflammation will stay for a longer time because it will mimic as if there is an infection. So immune cells will come there. So what we saw, in similar to what you saw in the TSA treated 1 micromolar concentration, you see no blastema. Same way, LPS treatment also, you see a decrease in the blastema. So the blastema levels are on par with TSA treatment. So TSA treatment also prevents the anti-inflammatory environment. It always favors the pro-inflammatory environment, and the LPS also favors the pro-inflammatory environment. So same effect is seen with blastema formation, as you can see here. In different time, when you go further, you can see the TSA treatment, how the blastema is low. The LPS treatment also, the blastema remains low. Here you can see the same thing we further added with another compound called N-acetylcysteine, which is a free radical scavenger. So free radical scavenger, basically, whenever there is a tissue damage, there will be a lot of reactive oxygen species. And whenever there is a LPS treatment, there is a lot of free radical will be formed. But if you scavenge them, the immune system will be told not to respond. So what we found, that if you prevent the reactive oxygen species from doing its job is what? attracting the immune cells and making the environment pro-inflammatory. So what we did was, you can see here NAC plus minus plus minus plus minus. That means in each experiment, control, we have two scenarios, with NAC, without NAC. TSA also with NAC, without NAC. LPS also with NAC, without NAC. But with LPS, with NAC, it rescues. And same way with TSA, with NAC, it rescues. But without NAC, with TSA, without NAC, with LPS, the blastema formed is minimal. So what it tells you, that NAC is acting as a scavenger of the free radicals. So it will sequester away the free radicals and allow less immune response in that site, in that spot, so that the pro-inflammatory environment will not be as robust, which favors the blastema formation. Same thing you can see the collagen deposit also. You can see because of the NAC treatment, the collagen deposit is not going as high as what it should have been, because the immune cells have a strong role in the deposition of the collagen. And this NAC rescue and restore the Treg levels also. You can see here DMSO and TSA in the caudal fin. This is the regulatory T cell levels, not much. This is NS, non-significant. But TSA with NAC, TSA reduces the Treg levels, which we know. But if you put NAC, that means the free radical scavenging happens. That means less immune response, then less collagen also. Then the Tregs are more present in the injury site. But NAC alone, it doesn't do anything, because it has to have TSA, so that more of the collagen deposit has to be there and more of the pro-inflammatory environment has to be there. Then NAC comes in rescue. In other words, if you are not hungry, whatever food I am giving will not look tasty for you. But if you are hungry, even my food is not that tasty, you will find it tasty. Same way, TSA makes a pro-inflammatory environment, creates less of Treg population migration there, but NAC allow more of the regulatory T cells, which restores not only Treg levels, but also allow the regeneration to continue properly. So in this proposed mechanism, what you are seeing here, this is the resting state, unamputated state. You can see acetylation in certain residues, the histone acetylation is there. And what happens is, when histones are acetylated, a set of genes will be turned on, and a set of genes which are off, they will not have any great role. So the homeostasis is maintained. But if you treat trichostatin for 3 days, what will happen? Acetylation levels will stay strong and high. There is no deacetylation is happening. And because of this, several pro-inflammatory cytokines and extracellular matrix genes are going up, because acetylation favors gene expression. And that is what you are seeing here. But if you have a uncut scenario, if you expose them with TSA, acetylation levels are high, along with this ECM genes and pro-inflammatory cytokines, you have abundant deposit of the ECM proteins, as you can see in this film. Remember, it is uncut, and now you are cutting it. When you are cutting it, remember, there is abundant collagen and other matrix proteins are already there in the fin, and the cells are not able to migrate effectively. And if you treat them with TSA, and if you rescue it with NAC or collagenase, then what will happen? You will get the MMP levels and other anti-inflammatory cytokines comes into normalcy, and you will end up getting a proper anti-inflammatory response, so that there will be a delayed regeneration that you can see it, and or normal blastema formation we can see it. So this information tells us how plastic the system is, and we can learn more about this regeneration and the Tregs in the next class. Thank you.